Hi, Professor Saho. I came across your ADNet paper on Boolean network modeling for Alzheimer's disease. Instead of just reading it, I reproduced the entire pipeline from scratch on my local machine using the public Bolad benchmark. This is the walkthrough of what I built, what I found, and one analysis I added on top. Cell 1 sets up the environment. Bone, Higmon, and ADNet are cloned from GitHub. And the working directory is pointed at the local Bolad data. The pipeline was originally built for the lab server, so I patched the IBD analysis to use a local explorer.conf instead of the hard-coded server path. 8 compatibility issues in total, all documented in the GitHub repo under patches. Environment is ready. SEL2 extracts the core output of the ADNet training procedure, the 40 gene bone Boolean signature. 20 genes downregulated in Alzheimer's disease, including NRN1, RGS4, and TSPA and 7, which are associated with synaptic function and neuronal maintenance, and 20 genes upregulated, including NOTCS3, NFKBIA, and NES, reflecting neuroinflammation and structural remodeling. This is what makes bone compact and interpretable compared to signatures with hundreds of genes. Cell 3 runs AD training all. This took about 3 minutes, so I'm showing you the pre-computed output. The bone signature is evaluated against 24 published AD signatures across 3 training databases. Bone achieves AUC of 0.82 on Friedman 2017. And here is the training performance plot. Consistent performance across all 3 datasets. Cell 5 is the full benchmark AD validation all. This runs across 22 held out datasets and took about 20 minutes. So again showing pre-computed output. One 1600 comparison rows against across 69 conditions. Bone is evaluated against all 24 published signatures on data it has never seen. Bone maintains AUC of above 0.70 across the majority of held out conditions, outperforming most competing signatures. This means AUC is around 0.82. This is fraction overlapping with bone signature, and this is the bone score distribution. This is the new analysis I added, not in the original paper. The Miller 2017 dataset is unique in Bolad because it profiles the same 90 donors across 5 brain regions. This lets us ask a question the paper doesn't answer. Does the bone signatures generalize across brain regions or is it region specific? Here are the results of the 4 regions: temporal cortex, parietal cortex, hippocampus, frontal white matter. The three gray matter regions are consistent at 0.74. Frontal white matter drops to 0.57, barely above any chance. This is cell 12. This is biologically meaningful. White matter is predominantly oligodendrocytes and myelinated axons, very different cell composition from gray matter cortex. The bone signature was trained on cortical data and captures gray matter diseases biology. It doesn't transfer to white matter. This directly motivates a white matter specific Bolin signatures as a concrete next step. This is cell 13. Sorry, the cell, as you can see, the cell number 13 prints the heat map. PEP compares bone against two other published signatures across the same four regions. The white matter drop is consistent across all three signatures, confirming this is a biological effect, not a bone specific limitation. And the cell number 14 prints all the generated files, training, and validation results. So I have added the full code base, all fixes documented, and this video linked in the email. Thank you for making ADNet open.